Hey guys, this is Kendall Terry, and this is a discussion on the three organs of a plant. Now we're going to do some things in class to really go more in detail with those, but this will at least give us some background information to build off of. So the first thing that we're going to look at is the first organ, which is the roots. What you'll find is these organs are pretty self-explanatory when we talk about plants, but when you look at a root, there's some identifying features here that we need to look at. First is that every root has a root cap on it, and that root cap is made up of a very thick cell-walled cells that are able to push through that dirt. You got to think about the root tip and what it has to push through for a plant. It could be pushing through rock. It could be pushing through the cracks of certain substrates or whatever. It may be pushing up against glass at times or whatever may be down there in the soil. So it has to have some kind of tip that will help protect it. That's what you see here. Notice shortly behind that you'll find the apical meristem cells. Those will be the cells that will be responsible for making all of the cells in the root and really pushing this root down into the ground. So the first area, the root cap, which is the protection to the apical meristem. Then you have the zone of cell division right here on the upside of the apical meristem, which cells will divide every 12 to 36 hours. So these cells right in here are all just dividing and dividing and dividing and dividing and dividing. And as they do that, they're going to help push this root cap down into the soil. Then you've got the zone of elongation. Those will be where cells will become longer than wider. So as they begin to stretch out, that's going to push those roots, that root cap down even further into the ground. And finally, you've got the zone of maturation, and this is where, in this area, is where you'll see the tissues develop, the specialization of those root cells. You'll see that'll be the development then of the vascular tissue, the ground tissue, the dermal tissue with the root hairs. All of that takes place here. If there's any kind of mutation going on, you'll see development happening down in these areas, which is not where those tissues are supposed to develop. They should be developing up there in the zone of maturation. Then you have an interesting fact of the root cap, or the root, not the root cap, but the root itself, is its responsibility is to take in water and nutrients from the soil. As it takes in that, you want to ensure that everything that comes into the vascular system, which is the highway that goes all the way to the leaves then, if it comes into the vascular system, that it's good for the plant. The thing that determines that is the Casparian strip. The Casparian strip is a lipid strip that separates the vascular tissue from the dermal tissue in the root. So you'll see it right here. And as things are coming in through the root, through the epidermal tissue and that sort of thing, dermal tissue, it has to get to the vascular tissue to go up to the leaves. So it has to go through these cells. It can't go beside these cells. It blocks transport between cells. Some transport will take place out here between the cells and not actually in a cell. But here at the Casparian strip, it cannot go between these cells. It has to go into this cell and then from this cell into the vascular tissue. And that ensures that everything that comes into the plant has to go through a semi-permeable membrane. It has to go through some kind of transport protein possibly, or it has to be small enough to go through the membrane just by simple diffusion. Whatever may be the case, but it has to go through that selectively permeable membrane of a plant cell. And this protects the vascular system, and it doesn't allow stuff to get into that vascular system that if it made it up to the leaves would cause major problems. Next we have the second type of plant organ, the stem. In the stem we have different internal stem structures. There's two different types here, the eudicots and the monocots. Sometimes this is just simply called dicots, but the more common name now used is eudicot. You can see that there's an obvious difference between the way they're set up. These are woody stemmed plants. Okay? So you've got the inside here, it starts in the end and it goes out. Of course, you've got here the vascular cambium, which is also referred to as just the meristem cells. And as they make xylem and phloem, it will help get thicker, or the diameter will grow of the plant stem. So you have primary phloem and primary xylem. Notice that's the furthest ones away from each other because as cells are made, it will push those away. So then you've got secondary xylem, secondary phloem right there around that vascular cambium. and then surrounded by that epidermis. Here in the monocots, notice that it doesn't have this circular pattern. Instead, you've got these groups of vascular bundles, xylem and phloem bundles, all throughout the stem, surrounded by a bunch of ground tissue. Much different look, and this is there's some distinguishing features of monocots, this being one of them, that the vascular bundles are just grouped together like this, as opposed to eudicots that actually have the rings that you think of with like wood that maybe you've cut down, you've counted the rings on the inside. That's actually what you're looking at, is the xylem rings that are developed that year. Some of the internal structures of wood, this is the best drawing I could do, sorry. So this is like the outside of the tree, and we're going to work our way in by layers here until we get to the inside. So the outside, you got the outer bark. Of course, you know what that is. Cork cambium, this is a layer, this blue layer right here, is a layer of cambium cells. It's the second layer of cambium cells that's responsible for growing the bark, the outer bark area, and the cork area that protects. Right inside that is the phloem. All three of these are actually linked together to include what we traditionally just call the bark of the tree. These are very thin. I know on here I've got them drawn thick, but it's just so you can see them. But these are actually very thin layers. Then you've got the vascular cambium ring here, the green ring. And then finally you've got the sapwood, which is the working part of the xylem, and the heartwood in the very middle, which is the non-working part of the xylem. So down in here, these xylem cells really aren't functioning. These are the xylem cells that are functioning. Then you've got the vascular cambium, phloem, cork cambium, and finally bark. Or the outer bark, I guess you could say. Alright, the last two here deal with leaves. In leaves, these are the primary sites of photosynthesis. Here's a little drawing of a cross-section of a leaf. You've got the cuticle on the surface, the waxy material that helps protect against evaporation and heat and different things there, water loss. Upper epidermis cells, the palisade mesophyll cells, you can see how those look different. The spongy mesophyll cells, and then you've got the xylem and phloem that's got a sheath of cells around it. Then down the bottom in the lower epidermis, you've got guard cells that surround the opening of the stomata. Okay? I'm going to go to the next slide that's really going to explain some of these. You can always back it up to look at the picture as we talk about them individually. So the last one here, we've got the cuticle. I already talked about that. That's that waxy or fatty layer on the outer wall of the epidermal cells to help prevent evaporation of too much water and water loss and different things like that. The palisade mesophyll, which is primary photosynthesis, those big, tall cells, those are where the majority of photosynthesis is taking place. They're crammed full of chloroplasts. The spongy mesophyll, those big cells, they kind of fit odd-shaped together, and it creates these air sacs for gas exchange. So there's areas inside the leaf for air to actually exist and wait to be used or sent out of the leaf. And then you've got the stomata, which is the opening that's surrounded by guard cells to open and close so that gas exchange can take place.